Hello all. In the previous session, we had discussed about one important concept of this chapter called replication, right? And in today's session, let us study about another important topic of this chapter called transcription. See, the name itself is suggesting about the process or mechanism of this process. The meaning of this very word you need to understand here. See, scribbling, you have heard a common English term called scribbling. You know what is scribbling? Scribbling means writing something randomly. ಏನನ್ನು ಗೀಚ್ತಾ ಹೋಗೋದಕ್ಕೆ ನಾವು ಸ್ಕ್ರಿಬ್ಲಿಂಗ್ ಅಂತ ಕರಿತೇವೆ, ಓಕೆ? ಅಥವಾ ಫಾಸ್ಟ್ ರೈಟಿಂಗ್ ಈಸ್ ಕಾಲ್ಡ್ ಸ್ಕ್ರಿಬ್ಲಿಂಗ್ ಅಂತ ಕರಿತೇವೆ, ರೈಟ್? ಸೊ ಹಿಯರ್ transcription or transcripting means what? Transferring the information from somewhere and scribbling it down somewhere else. You are transferring the information from one point to another point and you are writing it down by transferring that information from one point to another point or from point A to point B. That is the actual meaning of what? Transcription. I hope you are getting an idea now. So, in this process of transcription, at the molecular level, the transfer of information is taking from where to where? I have written here, the process of transcription means transferring of the information from DNA onto the RNA. So, how the information will be transferred? And the information is in what language? You know the language of the DNA. You know the language of these master molecules such as DNA and RNA. That will be in the form of nitrogen base sequences. Yes? So, this information that is present in the form of nitrogen base sequence will be transferred from DNA to RNA completely based on what? The principle of complementarity of base pairs, right? You know the principle of complementarity, right? So based on that, imagine, see, this is the strand of DNA. You know the DNA will be double stranded, yes or no? These are the two strands of the DNA. This is the open helix of the DNA. Both the strands are separated, and imagine some random sequence here. I'm going to take some random sequence. Whatever the sequence you have to take, you have to play with only 4 letters because only there are 4 nitrogen bases: A, T, C, G, C, G, A, T, C, G. Okay, some sequence I have taken, and you already know this sequence should be what? Complementary to the other sequence of the DNA or other strand of the DNA. So T, A, G, C, G, C, T, A, G, C. This you very well have already understood, right? So in this process, what is happening exactly here is that here the synthesis of RNA takes place based on the complementary base pairing. See, transcription means it is not only the transferring of the information from DNA to RNA. In fact, there will be no RNA at all. RNA iruda illa. RNA ಹೇಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, RNA will be produced just by copying the information. See, RNA nucleotides are brought together. You know already what are nucleotides, right? So nucleotides are carrying specific nitrogen bases, right? So here base pairing takes place, but the only difference is that in the case of the DNA, there will be thymine in DNA, right? Whereas the thymine is replaced by uracil in RNA, right? This point you very well know. Now here, if you consider this as the template strand, you know what is the template? Yes, template strand of the DNA is a strand which provides an open sequence for base pairing for the synthesis of a new strand. Maybe it is a DNA or RNA. Replication alli kaltideve navu, where there are 2 new strands of the DNA are formed, one from this strand and other from this strand, right? But in the case of transcription, the information is copied down. See, here in front of A, U should come, you know, because T is not there, and here C comes, G comes, C comes, again G comes, A, U, like this. This sequence what is being formed here, this is the sequence of the RNA. Okay, this is also being formed in only one direction, that is from 5 prime to 3 prime, right? See here, this is 3 prime to 5 prime, this is 5 prime to 3 prime. They are anti-parallel to each other, yes or no? And they are also complementary to each other. So this form or this process of copying down the information from a DNA sequence onto RNA, meanwhile biosynthesizing the RNA molecule, is called as transcription. I hope you are getting my point. DNA molecule in the one of the strands of the DNA. Keep that in mind. Always only one strand of the DNA, not both the strands. Why not both the strands? I will tell you that point. I'll come to that point. Okay. See, the process of copying the information from one strand of the DNA. There are two strands of DNA, but here only one strand of the DNA is used to copy down the information onto RNA. Meanwhile, while copying down the information from DNA itself, the synthesis or biosynthesis of that RNA molecule also will be taking place, and this very process will be called as what? Transcription. Yes, I hope you got an idea what exactly is the process of transcription. Of course, you are not into the details of the transcription yet. We will understand it now. Right? There are two main things that we need to keep in mind. One thing is that here, while transcription happens, transcription and replication, of course, in both the cases, both of these processes, they work on the same principle of complementarity. In the case of replication also, there are two newly formed daughter strands of the DNA which are produced from two complementary strands. Two complementary strands and this will also produce one new strand, this will also produce one new strand on the basis of complementarity, right? But in the case of what? In the case of transcription, only one of the two DNA strands will act as the template strand, and the other will not code for what? The synthesis of the RNA. So this is one thing that you have to keep in mind. The other thing that you have to keep in mind is while complementarity base pairing occurs, here, in the case of T, in the place of T, U will come. Thymine is replaced by uracil because it is a sequence of RNA. Yes. So now, why? Why is that? Why only one? That is that one strand. Why only one strand? So we need to answer this question. Why? See, if in case ಇಲ್ಲಿ ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಆಫ್ DNA ಇದೆ, both the strands are able to produce one molecule of RNA each, then the process will be faster, yes or no? ಈಗ ಇದು ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಇದೆ, ಹೌದಾ? ಎರಡು ಒಂದಕ್ಕೊಂದು ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಇದೆ, and there are already two strands of the DNA, and if this produces one new strand of the RNA, and if this produces one new strand of the RNA, the process of transcription or process of production of the RNA will be much more faster. Yes or no? But still, why the system is producing only one RNA molecule? Why only it is making use of one of the two strands of the DNA? It is because there are a few complexions here. There are a few complexities here, not complexion, complexities. See, what happens here is that now you know that these two are complementary strands of the DNA. Yes or no? And if this is producing one RNA molecule and this is producing another RNA molecule, okay, I'm going to draw it here. See, these two are the DNA strands. Okay, this produces one complementary strand here and this produces one complementary strand here. Imagine these two are what? Newly synthesized RNA molecules. Andre ಎರಡು ಸ್ಟ್ರಾಂಡ್ಗಳು, we are just this is just an hypothesis, this is not happening actually. ಎರಡು ಸ್ಟ್ರಾಂಡ್ಗಳು ಇನ್ವಾಲ್ವ್ ಆಗುದಿಲ್ಲ, ಯಾಕೆ ಅಂತ ನಾವು ತಿಳ್ಕೊಳ್ಳೋಕೆ ಪ್ರಯತ್ನ ಮಾಡ್ತಾ ಇದ್ದೇವೆ. Okay, so if both the strands of the DNA produce one new molecule of the RNA or one new strand of the RNA, or transcription is occurring on both the strands of the DNA, in that case what might happen is what we are trying to understand now. So this will produce this one, this will produce this one. So this is complementary to this and this is complementary to this. alva. So this one is complementary to this one is complementary to this. Let us see how it is. See, I'm going to write A T C G C T A T, okay, and complementary to this one. See, these two are complementary, right? I'm going to write the sequence on this one. Illi A ide T A G C G A T and A. Yes. Now this is producing a complementary RNA molecule because transcription is occurring. The only thing is that when there is A, U should come, right? U will come, that is uracil will come. See here U A G C G A U A. Here A U C G C U A and U. Okay, now try to compare these two strands. Of course, these are being produced in the 5' to 3' direction. 5' to 3', 5' to 3'. Now look at only these two RNA strands. Are they not complementary to each other? Look at this UA, AU, GC, CG, GC, AU, UA, AU. Are these not complementary to each other? Then there is a risk of why. One answer to this might be, so if these two strands are produced, then the two strands of RNA might form base pairing and become a double-stranded RNA molecule. If it becomes a double-stranded RNA molecule, there is no scope for protein synthesis. RNA production yak ಮಾಡ್ತಾ ಇದೆ ಅಂತ ಹೇಳಿದ್ರೆ, so try to recollect from the central dogma of molecular biology. DNA, it gets replicated, DNA will produce the RNA. That is what it is doing now. When RNA is produced, RNA has to be always single stranded. There is some sequence on this RNA, and that sequence that is present on the RNA will act to produce a structure of the protein by bringing about the sequence of amino acids. ಯಾವುದೇ ಒಂದು ಪ್ರೋಟೀನ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ, imagine this is the RNA molecule, and RNA molecule will be having a particular sequence of nucleotides, and that sequence of the nucleotides present on the RNA is deciding the sequence of the amino acids and the type of the protein produced. In order to produce a protein, this RNA should be single-stranded and the nitrogen bases on that RNA molecule should be exposed. If at all, if it is going to form a double-stranded molecule like this, ಎರಡು RNA strandಗಳು produce ಆಗಿ, if it is going to form a double-stranded RNA molecule, then it becomes a stable molecule. There will be no exposed surface or exposed nitrogen bases, and there will be no chance for protein synthesis. This is first complexity. Why only one strand is being used out of the two DNA strands for transcription process? This is first complexity. The second complexity that we need to look at here is that here the second complexity, this is first one, the second complexity I am trying to tell you here is that when two strands are being produced, okay, this is complementary to this one and this one is in turn complementary to this one. ಎರಡು ಒಂದಕ್ಕೊಂದು ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಇದೆ. That does not mean that they are same. The sequence on this is not the same as this one. They may be complementary to each other, but they are not same. That means this sequence may bring out one protein and this sequence may bring out another protein. Right? So here this complexity will actually cannot be dealt together in the system of the cell in order to bring different proteins at the same time. ಇದು ಒಂದೇ ಸಲಕ್ಕೆ ಎರಡೆರಡು ಡಿಫರೆಂಟ್ ಪ್ರೋಟೀನ್ ಅನ್ನು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡುವಂತಹ ಕಾಂಪ್ಲೆಕ್ಸಿಟಿ, this will be created because of the production of two new strands. Okay, so this will be having a different sequence, this will be having a different sequence, right? So it becomes impossible, even though if they do not come together and form a double stranded DNA, even though if they remain to be the single stranded RNA molecules, idu separate ಆಗಿ ಉಳಿತು, ಇದು separate ಆಗಿ ಉಳಿತು, ಇದೊಂದು single stranded RNA molecule ಆಯ್ತು, ಇದೊಂದು single stranded RNA molecule ಆಯ್ತು ಅಂತ ಹೇಳಿದ್ರೆ, the protein produced from this may be X and this may be Y. This is completely different and it is completely different. Right, so this is a much complex system it is going to become, and the cell system of the prokaryotes or even the eukaryotes presently, so as per our understanding, as per the understanding of the scientific community today, so this cannot be afforded by the complexity of the cell system as per today's understanding, right? So the second reason is that what? So the two different RNA strands, two different RNA strands might produce two different protein molecules, or they might code for two different protein molecules. Produce ಅನ್ನೋದಕ್ಕಿಂತ code for ಅಂತ ಹೇಳೋದು more appropriate. Okay, so this is the complexity behind the process of transcription. This complexity explains us why only one of the two strands of the DNA is involved in the process of transcription. Okay, why not two strands? I hope you have understood now, right? Moving on, now let us study about the transcription unit. So what is this transcription unit? Transcription unit is a segment on the DNA where exactly the transcription occurs. See, we just saw that one of the two strands of the DNA, there are two strands of the DNA, only one of the two strands will act as the template strand, right? The template means, you know, which provides an open sequence for copying the nitrogen bases and on the basis of complementarity and to produce the new strand of the RNA, right? So if at all, if the template strand is there, if the template strand is identified, then whether all the template strand will go on producing a new molecule of the RNA to elongate stretch, definitely not like that. See, look at this diagram. This is the DNA and this is opened up like this for transcription. Okay, now let me write the polarity. Let me take this different with a different color. This is one strand and the other one is one in yellow and the other is in orange, right? The orange strand, the what is that? Polarity of this particular strand is 3' to 5', right? Whereas the polarity of the yellow strand is 5' to 3'. Now, which one can be the template strand? That will be decided by the enzyme which will catalyze the synthesis of the RNA. Now replication ಕಲಿಯುವಾಗ we had learnt that the DNA synthesis, that is the new DNA strand synthesis takes place with the help of the enzyme called as DNA dependent DNA polymerase, and we also had studied about that particular enzyme, that DNA dependent DNA polymerase always works or functions specifically in one direction. Which direction? Try to recollect. Yes, 5' to 3' direction. Similarly, here we have got another enzyme called as the DNA dependent DNA polymerase but RNA polymerase. DNA dependent RNA polymerase. So this DNA dependent RNA polymerase is the enzyme which catalyzes the process of transcription. That means, in other words, I can say the DNA dependent RNA polymerase is the enzyme that is responsible for the catalysis or synthesis of the new RNA molecule, and this enzyme functions only in one direction, that is 5' to 3'. ಈಗ 5' to 3' ಡೈರೆಕ್ಷನ್ ಅಲ್ಲಿ RNA ಸಿಂಥಸಿಸ್ ಆಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ಇದಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಗಿರುವಂತಹ DNA ಸೊ ಆರ್ ಎನ್ ಎ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಲಿಕ್ಕೆ ಡಿ ಎನ್ ಎ ಅಂತೂ ಬೇಕಲ್ವಾ. ಸೊ ಡಿ ಎನ್ ಎ, ಇದು ಡಿ ಎನ್ ಎ ಇದೆ ಅಂತ ಹೇಳಿದ್ರೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಟು ದಿಸ್ ಒನ್ ಆರ್ ಎನ್ ಎ ಕ್ಯಾನ್ ಬಿ ಪ್ರೊಡ್ಯೂಸ್ಡ್. ಸೊ ಇಫ್ ದಿಸ್ ಆರ್ ಎನ್ ಎ ಹ್ಯಾಸ್ ಟು ಬಿ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಇನ್ 5 prime to 3 prime direction and this DNA has to be ulta, that is 3 prime to 5 prime, anti-parallelity, that you already know, right? That means the template, because the RNA synthesis always takes place in this direction, because this enzyme cannot function in any other direction, that is in 3 prime to 5 prime it will never function. It will function only in 5 prime to 3 prime direction, and that is why the complementary strand or the template strand to produce that kind or that polarity of RNA molecule has to be 3 prime to 5 prime. So this is the template strand now. So template strand. So out of the two strands of the DNA, ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಗಳಲ್ಲಿ ಯಾವುದು ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡು ಅಂತ ನಾವು ಕನ್ಸಿಡರ್ ಮಾಡಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ, the one which will be consisting of, which will be having 3 prime to 5 prime direction of polarity. Why? Because it has to produce the RNA molecule, or it should provide the complementary basis for the production of the RNA molecule or for the process of transcription in the direction of 5 prime to 3 prime. Yes, imagine there is some sequence here, A, T, C, G, G, C, T, A. So the same sequence, that means complementary sequence. So A ಮುಂದೆ T ಬರೋದಿಲ್ಲ, instead of T U will come A, G, C, C, G, A, U. So this is how it will be produced from 5 prime to 3 prime, and the catalysis, that is this process of synthesis of the RNA and copying down the information from DNA, the template strand of the DNA onto the RNA, will be catalyzed by this particular enzyme. While we talk about the specific action of the enzyme, you have already learnt about the enzymes in the previous year in the chapter of biomolecules, if you can recollect. Enzymes are always specific in their action, and enzymes cannot just go and bind to any substrate complex, any substrate and form a substrate, enzyme substrate complex. You have already studied about the lock and key hypothesis of the enzyme. The enzymes are very much specific in their action, and while forming the enzyme substrate complex, they will choose an appropriate and specific substrate. Yes, and here also, see, if at all the transcription has to happen, if at all this process of transcription and the synthesis of the RNA in 5 prime to 3 prime direction has to happen, this particular enzyme should go and bind to the template strand, and it should go on reading the information present on the template strand in the form of, that is, which is in the form of the nucleotides, and based on that complementary base pairing, the RNA will be produced. But in order to go and bind on the template strand, idukondu specific sequence bekalva. So the DNA dependent RNA polymerase, being a very highly specific enzyme, will require a particular sequence on the template strand of the DNA. Yes or no? Yes, it will require a particular sequence, and that sequence, the sequence to which the DNA dependent RNA polymerase can go and bind, the sequence on the template strand which provides the binding site for the DNA dependent RNA polymerase, will be called as promoter sequence. That will be called as what? Promoter sequence. I'll write it here. Promoter means what? Promoter is the sequence on the DNA template, the DNA template that provides a binding site for DNA dependent RNA polymerase. DNA dependent RNA polymerase. Did I say DNA polymerase before? If I have said DNA polymerase, please correct yourself. So it is not DNA dependent DNA polymerase, it is DNA dependent RNA polymerase. Please don't get confused. Illi baruvantaddu DNA dependent RNA polymerase, because in the transcription what is being produced is RNA, not DNA. In the case of replication, what is being produced is DNA, not the RNA. Alli baruvantha enzyme yaudu? In replication it is DNA dependent DNA polymerase. In the case of transcription it is DNA dependent RNA polymerase. Right? So you know, I hope you have understood, you are getting an idea what is a promoter sequence. Promoter sequence is a sequence, any random sequence, but a specific sequence which can be identified by the particular enzyme, and it will go and bind to that particular sequence. Antha one sequence which is present on the promoter will be called as what? Which is present on the template will be called as promoter. So promoter is the sequence on the DNA template that provides a binding site for the DNA dependent RNA polymerase enzyme. So once this enzyme goes and binds to this particular site, it goes on reading that sequence present on the template strand, which is in the form of the nitrogen bases. Okay, but until where? Whether it goes on reading until the end of that, it will never end, because it is a very long stretch of the DNA. But the proteins, see, you have already learned in the central dogma of molecular biology, if this is the sequence, if it is a strand of the DNA will be able to produce an RNA molecule, and the sequence on the RNA will be completely dependent upon the sequence on the DNA. Okay, so sequence of the RNA will in turn decide the sequence of the amino acids that are making a protein molecule. And there are many protein molecules, there are hundreds of protein molecules in our body which will be having different specific functions. And in order to make a specific molecule of the protein, a specific sequence of amino acids is required. Ade sequence of amino acids beku. And in order to get that particular sequence of amino acids, we need to have that particular sequenced RNA. So in order to have that particular sequenced RNA, we need to have a very peculiar sequenced DNA. That means, so this, which one? The RNA polymerase enzyme, it cannot just go on reading the sequence as long as it is possible. No. It has to stop somewhere, so that will be the particular sequence for a particular RNA, which will be later coding for a particular protein. That means, so it will go on reading on the frame of the template strand of the DNA as long as it meets another sequence of the template strand, which will stop the process of transcription. ಹೇಗೆ ಅಂತ ಹೇಳಿದ್ರೆ, see, this DNA dependent RNA polymerase, it goes on reading on this particular template strand until it reaches this particular strand, and that strand will be called as, it is called as terminator sequence. That sequence will be called as terminator. Illi hege ನಾವು promoter sequence ಅನ್ನು ನೋಡಿದ್ವಿ. Promoter is a small sequence. Some sequence is there. Okay, if this is the DNA, some sequence. Okay, so TATAAT one sequence ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. TATAT. So this is the sequence. Imagine this is the particular sequence which can be identified by this particular enzyme, and this sequence can be called as what? The promoter sequence. Right? So, and illi there are many nitrogen bases. This is the template strand, and there is one more sequence, like for example ACTTC ಇನ್ನೊಂದು sequence ಇದೆ ಅಂದ್ಕೊಳ್ಳಿ, and this sequence can be called as a terminator sequence if at all, if the molecule of the DNA dependent RNA polymerase will meet this, and on meeting that particular sequence, it gets detached. The enzyme molecule will get detached. Illi attach ಆಗ್ಲಿಕ್ಕೆ help ಮಾಡುತ್ತೆ. Which one? The promoter will help for the binding of the DNA dependent RNA polymerase, whereas the terminator will terminate or end the process of the RNA synthesis or transcription by detaching the RNA polymerase enzyme. So such a sequence will be called as terminator sequence. I am going to write it here. Terminator is a sequence on template DNA which terminates the process of transcription by making the RNA polymerase to detach from template strand. Okay, so that particular sequence on the template strand which will help in the termination or ending of the process of transcription is called as terminator sequence. But what is this sequence called? The sequence in between the promoter and the terminator will be called as structural genes anta kariteve. They are called as structural genes. Why they are called as structural genes? Because promoter ಕೆಲಸ ಏನು? ಇದು ಬೈಂಡ್ ಮಾಡುತ್ತೆ ಇಲ್ಲಿಗೆ ಬಂದು. ಓಕೆ, ಸೊ ಟರ್ಮಿನೇಟರ್ ಕೆಲಸ ಏನು? ಇಲ್ಲಿಗೆ ಬಂದ ತಕ್ಷಣ ಇಲ್ಲಿ ಡಿಟ್ಯಾಚ್ ಆಗಿ ಹೋಗುತ್ತೆ ಯಾವುದು? RNA polymerase. But in between, as it starts from here and goes on reading the template strand, so it is going to read the sequence present on the template strand, and by reading the template the sequence on the template strand, so complementarily the RNA molecule is being produced. The RNA molecule completely is determined by the sequence present on these genes or this particular segment of the DNA. And the structure of the RNA is completely dependent and it is completely determined by this particular sequence, and that is why this sequence that is present between the promoter and the terminator will be called as the structural genes. Okay, so remember, template strand will be always having the polarity that is 3' to 5' because the RNA synthesis can only take place in 5' to 3' direction. Because the RNA polymerase can only function in 5' to 3' direction. Yes, and there is a particular sequence on the template strand of the DNA which provides the binding site for the DNA dependent RNA polymerase, and such a sequence will be called as the promoter. And there is another sequence present on the template strand which will help in the detachment of the DNA dependent RNA polymerase and thereby terminating or stopping the process of the transcription or RNA synthesis, and such a sequence is called as terminator. Remember, these two can be very important definitions. And the sequence of the DNA, the fragment of the DNA that is present in between the promoter sequence and the terminator sequence will be called as the structural genes, because it is these sequences of the nitrogen bases will determine the structure of the RNA that is going to be synthesized. Yes, so this is how we learn about the transcription unit. So this particular unit will be called as transcription unit. Okay, but there is another strand of the DNA, right? So we were only talking about the template strand, and what about this strand? There is no function for this strand. The strand which is having the polarity 5' to 3' will actually have no particular function. It will not have any function, but still, very strangely, this strand is called as coding strand. Idu yavudannu code madodilla. There is no function at all for this one, but still it is called as coding strand, and this one is called as template strand. Actually code ಮಾಡ್ತಿರೋದು ಯಾವುದು? RNA ಅನ್ನು code ಮಾಡ್ತಿರೋದು ಈ strand, ಅದಕ್ಕೆ ನಾವು template ಅಂತ ಕರೀತೇವೆ. Whereas the other strand which is having the polarity 5' to 3' which is not coding for any RNA molecule, that will be called as what? The coding strand. Okay, and anything you refer ಈ transcription unit ನಲ್ಲಿ ನೀವು ಯಾವುದನ್ನ ಹೇಳ್ತಿರ್ಬೇಕಾದ್ರೂ ಕೂಡ, if you are talking about the promoter, if you want to say the exact location of the promoter, you will say the promoter is located at the 5' end of coding strand. That means you take the reference of coding strand. Where is the location of the terminator? The terminator is located at the 3' end of the coding strand. Yes or no? The promoter is located at the 5' end of the coding strand, and the terminator is located the 3' end of the coding strand, but it is not present on the coding strand, it is present on the template strand, right? So this is how you are going to take the reference with respect to the coding strand. This is about which one? The transcription unit, and we need to learn how to draw the diagram of the transcription unit as given in the textbook. I'll try to draw the actual diagram of the transcription unit so that you can understand, and that diagram is also very important for you. So this is the template strand. You know the template strand polarity will be 3' to 5', right? And the other one is coding strand, which will be having the polarity 5' to 3'. Yes, and here this sequence, some sequence on the template strand here will be called as the promoter sequence, and there is one more sequence present here at the 3' end, towards the 3' end of the coding strand. This is the coding strand. So this will be called as terminator, right? And this sequence is called as what? Structural genes. This is called as structural genes. So it is also called, the 5' terminal of the coding strand is also called as upstream. The 5' terminal of the coding strand is also called as upstream, and the 3' terminal of the coding strand is also called as downstream. Is also called as downstream. Remember, this is the coding strand and this one is the template strand. Okay, so this is the structure of the transcription unit. Transcription unit, which can be an important 2-mark question. Diagram matra kelidre 2 marks kelbodu, and if it is asked for 5 marks, you may need to write the diagram along with the explanation. Explanation alli en baritiri, what is promoter, what is terminator, what is the structural gene, what is the coding strand, what is the template strand. You need to explain each and everything here, including the polarity. I have told you very clearly, the polarity while writing the transcription unit, whenever you draw the DNA or RNA in this chapter, you have to pay attention to the polarity. Okay, 3' end il barutte, 5' end il barutte, that you have to keep that in mind. Otherwise the entire diagram, the entire concept that you are trying to explain will go completely wrong. Okay, so you please practice this diagram and also try to study this concept from the textbook.